Working for a change. Yeah. Oh, it's a bit cramped right back here, isn't it?
You put on the you're recording directly from a
Yeah.
signing into course.
Yeah, so should be recording.
Yeah,
I do it
see that.
automatic. I suppose the video's off. Do the sound. Yeah, should be fine. Oh no, camera control. Chat as well, comments if you want. I know you're not hearing it, aren't you?
Send you questions as we go then on your chat.
Yeah, so, when's it going to be over? Hi there. So, should I take registration then?
Oh, I can do it. I can take attendance.
All right, so we all here then?
Yeah, we're missing a couple, but they may have dropped the course. But
They're lost. Isn't
after last week's
Yeah.
too much work.
Well done. If you get rid of half a class, you're doing well. Oh, so we're good to go then.
Yeah.
All right. Yeah, well, some could. Okay, so I'm going to give like an overview. Like I can see most of you've done courses in food micro before. Did anyone do the principles of food science last semester?
Niran's in the FSQA program. This is my first semester.
Oh, so you've taken it. Oh, well, this is all revision then. So what I'm going to kind of do is go on more to do with concepts rather than facts and figures and things like that. You know, the way we think about microbiology and how we can use that to our advantage. There's a lot of slides here, as usual. So hopefully I'll get to the end. But like I said, I'm recording it. So if you want to go back and listen to it, you can, things like that. So what we're going to be covering is things such as evolution, how microbes came to be and how that's significant when we're doing control or trying to identify food, commodity and pathogen associations. I'll be revising the growth and basically what the different stages mean. Then I'll talk a bit about the pathogens and then spoilage, and then hopefully we'll get to the end. There we go. All right, so jumping already. So obviously, there's a long history in the Earth. We're 4.5 billion years old. Before that, obviously, we're kind of put together with interstellar dust from an exploding star and things like this. And there's different theories about how even the universe became established. The Big Bang theory, where they were saying it all started with this antimatter and matter going in and out of existence, and then we had a Big Bang. But not new theory, but another theory that's out there is that we're kind of just repeating. So we have a Big Bang, but then we start again. And that explains how life on Earth, I suppose, happened really quickly. So if we think about the Earth being 4.5 billion years old, how long in the history of the world, or when it first established, did we find the first life? So we'll say 4.5 billion is the start point. So how long into the history was it when the first microbe appeared? I should know this, it's common knowledge. Well, very short. About 350 million years, that's when we had the first common ancestor, as I say. So the life on Earth, there's different theories how it started. The first theory, which is, I'll try and get my marker going. No, it's not going to go that way. Oh, well, I'll just use that. The first theory is a tidal flat. So this is where we had this very pre-boiling soup, basically high in sulfur, high in ammonia. We had these tidal flats that went under wet and dry conditions, acid, alkali, obviously the UV of the sun and things like that. And that established the chemistry. What's that? We'll answer that. That established the chemistry of the building blocks of life. Now, the second theory is about life came from space. So Panspermia is where we got actual life forms coming from onto the planet, and people say it's crazy. How can you get microbes coming from space to land to establish themselves? But there's actually ongoing theories about, with Venus, for example, they suggest there's life on the cloud system, and they looked at asteroids and say, yeah, we've got some amino acids and fatty acids here. Neopanspermia is where we get the intermediates. So these are the building blocks. These aren't microbes coming from space, but amino acids and fatty acids and things like that. And like I said, they go to visit asteroids and comets and that, and they find traces of these, and people get excited. The third theory is that we're Martian, so life established on Mars whilst it had water and somehow got here, took a flight, so we're all Martians. Where we're going. Fourth theory is really what the common theories are, these hyperthermal vents, these vents at hot springs, putting ammonia, sulfur, hydrogen sulfide, and from these elements, building blocks such as amino acids, fatty acids arose. Then you get into much more similar theories about, well, it's all volcanoes, isn't it? You've got the heat, you've got nitrogen, sulfur, and all these elements to produce life. And then the sixth theory, this nuclear geyser, this sort of niche area where we had all the right elements involved. We had nuclear fusion because there was a nuclear kind of reactions going on. So the reality is we got all the building blocks of life. Some people say preformed microbes, if you believe that. But what do you think made them suddenly decide, okay, we need to make life now? Like, what was the point? Like, chemicals just exist, they react. But why do you think they wanted to become these self-replicating beings? Any philosophers here today, is there? Well, who knows? Who knows? But the reality is it did. And, people have got different theories how it all happened and things like that, and, what stimulated it to replicate. Now, we won't know, obviously, any of these because there's no way of knowing what happened all those years ago. But people have got theories about it. And the thing about life, I suppose you could argue, what's the definition of life? Is it living, metabolizing? Well, if it's that, it's not, viruses aren't involved. Is it has of nucleic acids? Well, prions replicate. So it's all these definitions of life. But anyway, to the point, to get on with it, isn't it? Is that people are debating what came first. Is it RNA or DNA? What would you say? Well, it's like you can be half wrong. Yes, so, Jasmine.
We think it's RNA.
Why do you say that?
Because it's more of a simpler molecule. It's only single-stranded, so it just seems like it would have been the first one to form rather than DNA.
I think you're half right there, isn't it? Yeah, people suggest RNA because, one, it can obviously carry nucleic acid material, which can then be copied to make copies of itself. It also has enzyme-like activity. And there's artificial enzymes out there, which people use RNA. So people suggest the RNA started, but obviously RNA is unstable if you ever try to extract any, and therefore DNA was much more stable. But people hang on to the proteins as well. When you think about proteins, they are more stable. They definitely have enzyme activity because proteins are enzymes, and they do reproduce in a way, and that's in the form of prions. So prions, we won't cover it, but just to know, now prions exist in two forms: normal form, abnormal form. And obviously they give abnormal forms give rise to CJD, variant CJD, and things like that. And it does this by contact. And so you could say, well, that's kind of reproduction as well. But anyway, the point is that we've got to the point where you say, well, no, RNA probably started, and obviously that encoded for proteins and life came on. So we've got all these elements of life, the amino acids, the fatty acids, carbohydrates, nucleotides, these building blocks of life. And there's a lot of prestigious scientists who try to recreate life. You can go back to the alchemist with the philosopher's stone. But there was an experiment done in the 50s, I forget who it was, Matthews or something, where they actually put all nitrogen, sulfur, a bit of methane, a bit of hydrogen, a bit of water together. And they produced amino acids and a few fatty acids from this. But interesting, with a chemical reaction, they got what we call a racemic mixture. So amino acids can exist in L or D form, whereas in nature, what's the main form that it persists? Oh, you could not have it. This one is the L form. So people are starting to suggest, well, that could be evidence that yes, it is preformed. But anyway, I'm digressing because as we normally do. So the point is that people are starting to figure out, saying, all right, we've got all these building blocks, but how did they interact to produce light? And obviously nobody knows. But what an interesting theory is about the responsive droplet theory. So what people do, or people did, is they produce these lipid vesicles as free fatty acids. I forgot which ones it is. And they looked at what we call the Marangoni effect. Not Alex Marangoni, the chocolate man. It's the Marangoni effect, where these vesicles were responsive because what was happening is that they alter the surface tension and they're directional. They'll direct themselves to sugars. They'll direct themselves to nucleic acids. When they encounter these nucleic acids, they envelop them. And what's more is that they start splitting off. So it looks like they're literally living. It's a parlor game, obviously. It's not real in terms of life, but it could be. So really what the vesicles did is give the environment for chemistry to happen. You're containing everything now. And what went from there is that the first viruses appeared. So what these viruses were doing is we're taking nucleic acid from one vesicle, transferring it to another. Now, the viruses, as they normally are very selfish. They wanted to replicate themselves, which they did. But they also carried over some nucleic acid material that encoded for enzymes. So what we had is this self-assembly lipid layers, which people say this is the secret of life. We had the proteins in there, and we had the nucleic acids. But obviously, they were really confined to the hot springs, if we go for that theory. And the good thing about springs is they're fairly consistent. You know what the temperature's like, you know what you've got a constant source of sulfur and nitrogen and carbon and things like that. But obviously they wanted to break free. So they started going away from the hot springs. And even to do this, they had to become independent of the hot spring and more dependent, be able to survive and metabolize. So increasing survivability really modeled evolution, and they needed to generate their own energy now because obviously they couldn't depend on the hot springs for sulfur reduction and things like that. And that's what kick-started evolution. So the first ancient bacteria were the archaebacteria, and these were our common ancestors. And they did lack this peptidoglycan. They had something like a pseudomeuron, which is a kind of primitive cell wall. But you can go out today and isolate them if you're willing to go to hot springs and things like these. And really what indicates that it could have been hot springs is these are what we call extremophiles. They like the hot temperature, they need the hot temperature, they need high salts to grow. And obviously methane, as we talked about, is being the building blocks. So these are still here today, and they kind of extremophiles of interest if you're trying to isolate enzymes or whatnot. But obviously, other microbes evolved and branched out. So one of the big sort of landmarks, if you want to call it a landmark, in evolution was the establishment of gram-positive and gram-negative bacteria. But which came first, the gram-positive or gram-negative? Don't talk very much today. Oh, yeah.
I would say gram-positive.
Well done. There you go. So yeah, there were well, again, people can put all these theories out, but nobody was there. There's no evidence for it. But the gram-positives were the ones that evolved first because they had to live with desiccation, fairly extreme environments. Now, the gram-negatives differ in that they've got a very thin peptidoglycan layer, but also what they have is outer cytoplasmic membrane. And this outer cytoplasmic membrane contains something called LPS. LPS, lipopolysaccharide, and this is what we call a superantigen. This is very antigenic. But really, the purpose of the outer cytoplasmic membrane is to give protection against sanitizers, antibiotics, and other diffusible antimicrobials that would be encountered. And you know who invented the gram stain, do you? Well, the name gives it away, Christian Gram, isn't it? And the way the gram stain works, obviously, is you have crystal violet and iodine, which form a complex, which can't penetrate the outer cytoplasmic membrane, but it can obviously stain the peptidoglycan and gram positives. And in a sort of gram stain, you use ethanol to strip out the membrane of the gram negative and then counterstain with staphynon. But as you know that anyway. All right, so with gram negative and gram positive bacteria, then obviously with the gram positives, there's always exceptions to rules, but generally speaking, they're more firmly resistant if we think about Listeria. The desiccating resistance, think about Staphylococcus aureus, whereas the gram negatives are much more tolerant to diffusible sanitized antibiotics and things like that. Now, the thing was, though, is that peptide cell walls are good, is essential to some case, but they do make the microbe prone to antibiotics. You know, a lot of antibiotics act on the cell wall. So you'll get these L-form bacteria which lack cell walls, and these make up a small proportion of the population. Like you can go sieving for them, it's only less than 0.1%. But without these cell walls, they become more resistant to antibiotics. The target isn't there. They're very slow growing, but they're very resistant. And these are hedge betting. This is basically a subpopulation that if all hell breaks loose in the environment, at least you have some survivors. And these require these hydroponic, very high salt to survive and are slow replicating. People put a lot of used to, not though so much now, L-form bacteria. Back in my day when we're doing a PhD in that, people talk about L-forms. This is natural environment and things like this, and these are what cause latent infections. And it's quite interesting, I'm digressing, I'll do that a lot, is that you've heard of the CRISPR system, for example. Has anyone heard of CRISPR? Well, you should do, Johnny, you thought it. Well, CRISPR system is this sort of bacteriophage resistance, because obviously you don't want phage coming in, disrupting everything. Very all right for lysogens and things like that. But what's interesting is that trying to prove the significance of CRISPR in a laboratory environment doesn't work very well. But if you get natural isolates, like there was a paper just yesterday published about E. coli and CRISPR, it's much more significant. So when people talk about food microbiology or microbiology in general, saying, oh, well, everything's been discovered, in reality, we're far away. So what you find in the environment is much more different. So as I said to you, the cell wall was a vital step of evolution because this gave rigidity, protection, because obviously you want to, if you're evolving, you want very stable genomes. You can't have genomes just changing every time because you go back to the beginning and go back to the start. So the cell wall gave that protection. And obviously it gives the shape and size of the bacteria. Now, why do you think, and probably no one's going to answer, why do you think we have rods and why do you think we have cocci? And which is the best form to be in? So why do we get all these different morphologies of bacteria? Yeah, I thought you would. What would you guess? So would you, if you were a bacteria, would you rather be a cocci or do you think you'd rather be a rod? No preference. Well, it's one of these mysteries of, like I say, you might think, oh, it doesn't matter. But in actual fact, in evolutionary terms, it does matter. So cocci have very high surface area ratios. That makes them much better at nutrient acquisition. So this is why Staphylococcus, for example, that lasts a long time because it's adapted to absorb in low nutrient environments and things like that. Whereas rods, you're much better at motility. You're much better at building biofilms and things like this. So again, it's one of these things that people suggest, but I don't know if you could ever prove it. But the important thing is that we could establish stable genomes now. Yeah, viruses and bacteriophage, like bacteriophage, would come and attack, but these are pros and cons. Bacteriophage transfer genes as well as replicating themselves. So I suspect, a small portion of the population have to be sacrificed. And it enabled bacteria to diversify, because they don't need the hot springs now. They can survive in lots of different environments. Now, evolution and adaptation are two, well, similar but different. So evolution is literally, as you probably know, is hereditary characteristics. So these inherit genes from one population to the other, and they build on those, and that's what evolution is essentially. That's why microbes branch out and things like that. And it differs from adaptation because adaptation is basically the phenotype. This is where you would adapt to a stress and things like that, or you might go dormant. And adaptation, obviously, is what we encounter in the food system. We also encounter evolution as well. You know, it's just the way we kind of characterize it. All right, so back in the day then, obviously, civilizations come, you know, if we came around 300,000 years, and I forget when Aristotle was doing his thing, I think it was 500 BC or something like that. And what they wanted to do, obviously, is try to explain the world, trying to characterize the world. Obviously, there weren't no microbes until Van Leeuwenhoek in 1600s looked under the microscope. Even then, they didn't say, well, it doesn't matter about those. It was only, I would say, 1860 when Louis Pasteur basically put forward the germ theory. Everything was spontaneous before that. So typically then, they only could see creatures they could see. They didn't have microscopes and things like that. And Aristotle was saying, well, you know, it's spontaneous. You get all these different animals, and those that survive have the required traits, and the others go extinct, which was, it's not far wrong in a way, because life must have been spontaneous if you believe about the hot springs and things like that. And it's true, you know, if you got the traits to survive, you will survive and prosper. But it was only till Charles Darwin came on board, and you've got to remember these years in the 1800s and before, was all ruled by religion, and anything that goes against the text, you know, you put your head up, a bit like going onto Twitter and saying the vaccines don't work or COVID-19 is not real, sort of thing. But Charles Darwin, it was a depressive kind of character, I suppose, but I suppose he would be. He went to the Galapagos Island, I think it was, and he noticed these different animals there. And he came forward with natural selection, survival of the fittest. He probably stole the idea from Aristotle in a way, isn't it? And that's the sort of hard and fast rule peoples live by. It's natural selection. Microbes survive for those who can survive and obviously exploit the environment. But certainly, people built on the natural selection idea, saying all life goes to complexity. So I guess we're the most complex, although I think there's a few others. But what they're suggesting here is that the branch of evolution develops, and we go to dead ends. Those dead ends are bacteria, yeast, and things like that. These weren't successful. But the branch of complexity is where everything tries to get to, which isn't really true, is it, when you think about it? Because if that was the case, we would have this selection where only a few beings would be here. But the idea is at the time saying everything drives to complexity. But then we started to look at different types of evolution. And there was a character, Hamilton. He was a social scientist, actually. He only produced 6 papers in his lifetime. I don't know how he managed to pass P&T, but there you are. But I'm sure he did well. So he was saying, as a social scientist, he was not a microbiologist at all, but his kind of philosophy or his theory was that we evolved to be cooperative, and we cooperate to make civilizations, but we cooperate with those which are genetically related to us, i.e., you'll do it for your family, you'll do it for your friends, but you wouldn't do it for total strangers. But this cooperation is at the cost of what we call the actor. So basically what we're saying there is that you would do something without getting anything in return, but at a cost to yourself. I think we've all been there, haven't we? So people have looked at this and say, well, yeah, it's true, isn't it? Because when we think about microbes, all we think about them is beating each other up, for one thing, but living as single cells. So if I'm a bacteria, I'm going to live by myself and just hopefully compete. But no, populations kind of get together to cooperate. So if they're genetically closely related, because you don't want to give advantage to obviously competitors or things that aren't related to you. So what the theory goes in the microbial world is that those that cooperate ultimately succeed in making bigger populations. So, for example, one cell might have antibiotic resistance. So if they gave it to all their kind of relatives, they become antibiotic resistant. So yes, they will thrive, but there's no advantage to the cell giving the antibiotic resistance, but that's how it works. But those that don't cooperate don't succeed. I suppose it makes sense, doesn't it? If you don't all act as one, you're going to be taken out. But pathogens have learned, and a few other bacteria obviously, is that you could exploit this by being non-cooperators, but exploiters, and you could exploit the sort of activity of others. And one of the big sort of significant parts or relevant parts is in biofilms. So we know biofilms, these extra polysaccharides with alginic acid, dextrans, and things like that. And these cause a clumping of cells and adhering onto the surface. Now, once they're adhered into these biofilms, then you can get homogeneous biofilms, basically one type of bacteria. I don't think it really occurs in nature. But you also can get pathogens associated with biofilms as well, which gives them protection, physical protection. They get genetic sort of exchange going on. They switch to dormancy, which basically means that they go into this dormant state, non-growing state, but very stress resistant. And you might have heard on the news about a campus, I forget what campus it is, Cape Breton, isn't it? Where they're having really big trouble with the water. Basically for the last 4 months, is it Cape Breton, Giselle, is it? No, it's all right. I think it's a university campus, and basically they've had coliforms in water and all these E. coli in water. And the reality is they've been going on for about 3 or 4 months now. And it was on the radio last week, I wasn't on the radio, I was listening to an interview by their head person. And they said a few coliforms in water isn't dangerous. I would suggest that it is. But what I'm bringing it up for is that this is this thing about biofilms. Once they're established, they're very hard to remove. It's not just a case of putting sanitizer down and things like that. They persist for a long time. Yeah, don't go to Cape Breton.
Yeah, it is Cape Breton.
Is it? Oh, there you go. What a genius. But yeah, they've been having big troubles. And, what they'll have to do is take all the piping out. I think they're reluctant to do that, but that's what they'll have to do. Anyway, to the point. So cooperation is cooperative evolution where microbes benefit from each other, making biofilms. If you try to make a biofilm with a single cell, it's a bit like trying to make a brick wall or a house. It's very hard, but if you've got all your kind of relatives helping you or your friends, then it's easy. So cooperation, public sharing, cross-feeding. When we go to the microbiome, the reality is that certain cells produce butyric acid, and butyric acid is the center part of cross-feeding. So one produces butyric acid, other microbes benefit from it. Then you get the cheaters, and this is where pathogens come in. They'll take advantage of all these goodies, these substrates or activities of the good cells and produce biofilms or incorporate into biofilms or maybe trigger virulence and things like that. Then we get the predatory bacteria. So these can be vampire vibrio, I think they call it. Yeah, vampire vibrio. What a name, isn't it? So these basically go inside cells and eat them. And the group hunting, I don't know an example of that, but it's good to think, isn't it? You've got all these microbes, getting these cells going around, ready to attack, isn't it? And then you've got ones like Listeria, that's got the Midas touch. So Listeria contacts a cell and it'll punch holes in it. So it's a pretty, cooperation to a degree. Sometimes cooperation can turn deadly. So the thing about this theory about going to complexity then, we know, yes, microbes do get complex and that, but the reality is that going to complexity means we can survive. So, for example, we can go out in this -20 weather, we can go out in the +40 when it gets to summertime, we complain about the heat. And that's the good thing about complexity, eukaryotics, they tend to be much more adaptable, but slow mutation. We can't adapt as quick as bacteria can and things like that. So evolution doesn't always go for complexity. Sometimes it can go to reductive evolution, i.e. you're getting rid of all those genes that you don't really need, and then obviously adapting to the host. So that's kind of reductive evolution. And the ones that do this are obviously the parasites. So viruses, you could say they're a parasite, even though they were there first. But things like enteric protozoan and even some bacteria like Shigella, they would lose traits to able to survive in the environment and basically acquire those that survive in the host. So these don't survive well in the environment, but they do very well in the host. So again, it's this reductive microbiology or reductive evolution. And when you become a pathogen to the host, you have to kind of short circuit its immune system and not activate it, because once it's activated, all hell breaks loose. So what a lot of pathogens do, which are parasites, like as I mentioned, toxoplasma, things like that, they try, and salmonella to a degree, is that they'll neutralize the immune system, they'll either hide from it, and then obviously go associated with the internal organs to become, the host becomes a carrier. So with the immune system then, you've got two types. You've got the innate immune system, which is basically active all the time. Physical barrier, skin, for example, T is in our eyes. We've got natural killer cells, complement factors. These are there to give us continued protection to a point. But then you get the induced hormonal immune system, which produce the T and the B cells. So what do T cells do? It says it, I can't even read that, but what does T cells do in the immune system? What do you think? Oh, you can't read it. Oh, well, I don't know, we'll read it even on. So what happens when we ingest a pathogen, if our own innate immune system can't take care of it, we've got all these B cells. These B cells can be random, it's got random antigen antibodies on them. And when they lock on to a target, salmonella, for example, they get the T cell to activate them. So the T cell comes along, releases cytokinins, our temperature goes up, we're starting to feel aching, we'll probably get a bit of nausea and probably a bit of diarrhea and things like that. So the body kind of does it. And the T cell activates the B cells to clone them. So you get this increase in B cells, you get memory cells, and T cells obviously interact with macrophages as well. The macrophages are those cells that go around the blood system trying to capture the foreign protein. Once it digests it, it presents it to the T cell and things like that. So there is a reason for this, as I'll talk about later. So we've got our immune system, and like I say, that what the parasites do, and pathogens in general, is develop their systems to avoid the immune system. After saying that, like Staph aureus, for example, wants to activate our immune system, so all hell breaks loose so it can get the iron from the blood and things like that. But typically, they want to lay low. So as I said, reductive evolution, people are suggesting, is that why viruses are so successful. You know, they go more simpler and simpler. There are macrophage viruses and things like that. There's this bacteria, which I can't remember the name of now, which is basically this intermediate between a cell and a virus. But anyway, we won't dwell on that. When you think about viruses, they are rapid growing. They adaptation through mutation. That's why we get all these different COVID variants. And there's a virus for every sort of microbe, every sort of animal. The only kind of different one is the yeast, which have a killer factor, which is an ancient virus. But let's not confuse things. So people are suggesting, oh yeah, reductive biology, this is why the viruses are so successful. Whereas others say, well, no, viruses just got stuck in the past and never evolved to be anything else. Yeah. Whatever you decide on. So to the point then is that we have different forms of evolution. You know, we have this drive to complexity, natural selection. We have this cooperative behavior, and we have reductive behavior. And you could say, well, complexity must be the most successful. But I think you're looking about 40,000 years' time and see who's still here. And then you can say, oh, yes, reductive was the way to go, wasn't it? All right. So as I said to you, we live in a microbial world then. You know, we came on very late in the game. The first life 350 million years ago. And what microbes have done is adapted specific niches. So what you'll see with bacteria is that they grow in a very small sort of a niche, you know, in terms of intrinsic, extrinsic factors, whereas the eukaryotics are much more broad, adaptable, but slow to adapt. You know, microbes can be on there, isn't it? And obviously we have these communities as well. So typically then, the thing that defines growth is the intrinsic, extrinsic factors. And we know the intrinsic factors are associated with the food or associated with the environment are things like nutrients. Are they available? Can they be metabolized? Things like that. Obviously, the pH, is it acid, alkali, or neutral? The redox potential, is it reducing environment for anaerobes, or is it oxidating environment where the aerobes can survive? The water activity, the available water, not the moisture content, but the availability of water. Some, obviously, bacteria like it fairly high, but you get some that are adapted, as we talked about the archaebacteria, which like it high salt. That's what they're adapted to. And even the food structure, are we on a solid? Is it a hydrocolloid? And things like that. All these affects the growth. And obviously antimicrobial agents, what's there to stop it growing and how can it overcome it? And then we have extrinsic factors such as temperature, which is an obvious one. That's why we can refrigerate things, stop things kind of growing. Is it anaerobic, aerobic? Is it relative humidity high? Is it low? And obviously the resident microflora. I always find it odd that it's listed under extrinsic factors, but that's, I suppose I had to kind of balance the thing. So I think it goes without saying, and you've probably cottoned on, is that these what we can use to either encourage growth of microbes, or try to enumerate them, but obviously inhibit the growth when they're not. And we also can use the intrinsic, extrinsic factors to look at pathogen food associations, which we'll talk about in a bit. So we can use this knowledge of intrinsic, extrinsic factors to do a hurdle approach. So the hurdle approach is that we've got a choice. We can do canning, anything sterile, don't worry about anything, but not many people like that idea. But what people do like the idea of minimal processing, clean labeling, if you want to call it that as well. And this is where we use a combination of hurdles, intrinsic, extrinsic factors, to inhibit different groups of microbes. So collectively, we don't, just one of them on its own won't, because obviously if we decrease the pH, for example, the lactic acid bacteria take over. But if we put a preservative in, like hop extracts, then we can stop the lactics growing, and we can put them under MAP and refrigeration. So the hurdle approach is a collection of intrinsic, extrinsic factors to inhibit the growth of microbes. But obviously, there's always one spoilage microbe that comes through. All right, so we got to the point then where we've evolved, we've adapted to a specific niche, but then we have to grow. Microbes have to grow to furnish the sort of evolution. And you probably came across a growth curve like this in high school or even the first year, where you say, well, we've got a lag period, we've got an exponential growth period, and then we have stationary, and then we have a dying off. Obviously, in reality, this is very seldom the case. Microbes probably could live in the lag phase for a long time. Obviously, the most of the time are in the stationary phase because they've got no nutrients around them. The conditions are pretty hostile. And whether there's a death phase, again, I think it's more like a roller coaster ride, that it's not straight. Very seldom do they find themselves with the ability to grow. Certainly in fermentations, we do get growth, but a very artificial situation, isn't it, compared to the environment. In the environment, they're competing for nutrients, and when they arrive, it's who gets there first kind of thing. So microbes do grow, but a lot of the time, they're kind of just resisting the stress and things like that. So as we know, the lag phase then is when there's lots of things goodies outside the environment. You've got all these nutrients, your pH is too bad. But you've got to have this period to kind of repair yourself. You've got to repair from all that stress you've been under. You might have to synthesize enzymes. So if you land yourself in milk, for example, with milk, the main sugar is what? Well, there you go, lactose. So you have to express
Taste.
yeah, on the lactoperon. Yeah, you're right. Yeah, you've got to express the lactoperon to get the all the enzymes required in the porine to transport them, because you don't want to express them all the time. You want to do it. So during this period, you're not growing. You've got nutrients out there, but you're getting the enzymes and transporters together to exploit them. Now, in a lot of food preservation, like for controlling listeria, for example, we want to extend the lag phase, because we can't kill it. If we want, we could kill it, but we need to have such treatments there, it would alter the product. So we could firmly process it, but then people say, well, it's these sensory properties do. And then you could add things like lactate diacetate, which controls listeria, but you can't add too much because it makes the product very bitter. So what we try to in a lot of ways, and listeria is one, but what's the other pathogen we try to extend the lag phase for? Well, there's a famous one, obviously there's others as well. Think about the most lethal pathogen we encounter. So what would you say is the most lethal pathogen we encounter? In France, for example, if you go to a restaurant, who just do their canning of their sardines by themselves. Okay, what is it? Oh yes.
Clostridium botulinum.
Oh, there you go. Steve, so yeah, Clostridium botulinum. And so what we try to do with low acid foods, and we'll talk about this when we come to food waste, is that we try to extend the lag phase, because we know once they start growing, they're producing toxin. When they produce toxin, obviously that's lethal. And there was an outbreak of, I think 2, maybe 3 Canadians died when they went over to France during the World Cup of rugby, I think it was. And they said, oh, we'll go to this restaurant. And they had home canned sardines. And obviously they hadn't been processed right, and you get botulism. Other ones like Staph aureus, for example, is another one. So I was involved in a project where they were making waffles and things like that. And they had batter. And they wanted to say, could we go for 5 days solid processing without sanitation? And we said, well, it's a long time to go without sanitation, because with batter, as you know, it's fairly neutral, obviously high starch, high nutrients. And yeah, basically they wanted to know when would Staph aureus get to grow and get to that dangerous phase. And Bacillus cereus is the same. But anyway, I'm digressing. So the point was, we tried to extend lag phases. And if you're in the Arctic, basically your lag phase could be a year, things like that. But you do grow. And like I mentioned to you, don't think of bacteria as homogeneous. People say, oh, all the cells are acting the same. There's a lot of hedge betting going on, i.e. a subpopulation will go dormant. They can grow, but they don't decide to grow, because if all hell breaks loose, then the entire population gets wiped out. And we call these persister cells. So persisters, like dormant cells, like VBNC, which is a bit different, we'll talk about later. But these are the cells that, and it's only about less than 1%, where if the antibiotic hits, they'll survive and the rest of the population get wiped out. So never think about populations as homogeneous. Even in chemostat cultures, people are starting to think, well, yeah, it's supposed to be homogeneous, but it really is. So we have our sort of anabolic and catabolic processes going on. Basically, anabolic processes are building up, anabolic steroids for those who go to the gym, or catabolic metabolism, which is producing ATP. Now, we have a lot of different growth rates when we look at different bacteria. The quickest one is Clostridium perfringens at 9 minutes. We've got some long ones, like Listeria has 45-minute generation time. But why do you think we get different generation times? For losses, Well, obviously, no. Well, people think they know, but it's complexity, isn't it? The more complex the cell, the more it has to grow. Even the metabolism, can it regulate the metabolism? Because it can't if you go metabolize too quickly, you overheat. And overheating isn't good, not only for your car, but obviously your cell. So this is why you have different generation times. But these become important when we try to predict the growth of microbes, which we'll might talk about in a bit. But then we get to the stationary phase. So in the stationary phase, this is where microbes exist. They haven't got nutrients anymore. They've all been converted to acids or things like that. And this is where you start getting to the point where you've got to start battening down the hatches or do something. And with stationary cells, the downregulate become VBNC to a point, or at least express the stress proteins, and this is where they spend most of their life. Now, when you get to the point of the late stationary phase or exponential phase, you've got a collection of your own bacteria, your friends around you, your relatives around you. And this is a time to start acting. This is your maximal population. Most of you are intact, unlike when you go right down the road in stationary phase and you start dying off. So this is the point where pathogens might say, let's launch the attack. Launch the attack on plants, launch the attack on a host or body. You might be saying to yourself, let's build a biofilm. We've got enough of our own around us to produce EPS. Let's get a biofilm made. We might start producing enzymes like proteases and lipases and things like that. Or we could decide, let's get out of here. Let's be a Proteus and swan to another area where the mutants are good. Or if you're Vibrio fischeri, you could start producing bioluminescence, basically to tell a would-be predator, say, look, we're here in this squid, come and eat it so we can transfer to you. And this is quorum sensing, this coordination. But what's the signal call for quorum sensing? Does anyone remember? Fair enough. Well, autoinducers. So the thing is, different microbes produce different autoinducers, i.e. signal molecules, because it's a bit like when you're in a war scenario, which I guess we always are, to communicate to your own. So basically, you send a signal to your own saying biofilm today, or send a signal to have an attack on the host, the virulence factors. And there's a whole range of different signal molecules, like Pseudomonas produce 36 signal molecules. And what pathogens do is that it can intercept these signals. So they've got all these signals. One of the signals will say, yes, we are our own kind. Let's do virulence. Let's go and attack the host. But then if we've got the signal from, say, for example, a Pseudomonas and say, well, this is our own, but it's a Pseudomonas, so there's no point in attacking the host. Let's form a biofilm. So it's literally like circuitry instructing what the pathogen should or should not do. And with the quorum sensing, as you probably know, it is a cell density effect. At very low cell density, you won't be any signals, they'll diffuse off. But when you get a high population, a high concentration of signal, that tells you things are going to start happening. And what some plants do, and what some people are suggesting, is if you artificially put in the signal molecule, you can get the cell to act, without all its population around. I.e., you can make it think that it's got high numbers, but in reality it hasn't. So you can take them out. And that's what plants do. Plants send out these signal molecules, thinking these plant pathogens will attack it. But because there's only one of them, their defense system can easily work. So that's quorum sensing. So as I mentioned to you, when you get to stationary phase, it's pretty bad environment. You're starving. All the nutrients have got to be converted to acids, maybe. You probably might have osmotic stress if you start desiccating. You're going to be under oxidative stress, so someone's put some bleach peroxide there. And obviously antibiotics produced by bacillus or moles will be there as well. So basically, microbes have to evolve this because this is a common situation. All the nice hot spring things are gone. It's a very changing environment. So some microbes decided to go the endospore route. So producing endospores, which can lay dormant for millions of years, if need be. Others start saying, let's build a biofilm, get in there and just hunker down until the next one. And obviously expressing the stress proteins. These are going to shore up the ribosomes and the DNA and things like that. And then it's this dormant state. So a dormant state is where the microbes downregulate, become non-growing. And when we say non-growing, sometimes become non-culturable. And there's a few different types of dormant state. L-form, people say, well, it isn't really dormant because they are slow growing. But the two main dormant forms are VBNC, viable but non-culturable, which the name suggests. They can't be cultured using normal media. They need something special, like our GI tract or something. And then the persister. So the history, I won't go through the history because, like I said, I'm looking at the time now. So persisters were of interest to clinical people because they were the populations that survived after an antibiotic addition. So during the war, whilst everyone was fighting, Bigger did this experiment where he got Staphylococcus aureus, exposed it to penicillin, The population decreased, but you always got this sort of subpopulation, and they call persister cells. VBNC, some people say, are cells that are just being beaten up and they're just on the verge of dying, but they could pull up given the right conditions. And, when I go to the IFP this year, they want me to talk about the significance of persisters in food systems. Again, should be interesting because nobody knows, but I'll work on it. So just to illustrate about it then, so there was a group in Japan who went in to get these sediments, and they revived these 100-million-year-old bacteria. It took them 1.5 years lag phase. So could you see the patients? Like I know a lot of you say after 24 hours, if it's not growing, it's definitely dead. But it took them 18 months lag phase to nurture these out, and then they just grew, and they were acting normal. So it really comes to the point, isn't it? Is where do you define death?
Did they use salt with that one? What was from the sea? Were they using higher salt?
I must admit, I think they probably did. Yeah, they would have done, because obviously they wanted to mimic it.
Yeah.
So I imagine it'll be like a salt solution. I don't think they were very inventive about the carbon source. I think they just put glucose in. But what was quite interesting is that they had the patience to go, and I suppose they expected it, because there was an experiment done a few years ago where somebody had these cultures. They got single cells, they say, in each well, and they just left them for a year, 2 years, and they had these would go positive every now and again, and that's a scalp theory. So basically, the theory goes is that some cells on a timer, don't tell me what the timer is, I've got no idea, is that they'll wake up and say, is it good to grow? And say, no, it isn't, we're going to die. But eventually, you hope that there will be, and that's what happened here. So really what it illustrates is the fact, it's probably contamination, isn't it? But what it illustrates is, how do you define death? And that's the key point. So typically in a normal culture, then what you have is actively growing cells, they grow up in population. It gets a bit nasty, but you can then subculture, where you inoculate subculture into another broth, and the cycle starts again. But then you get into what we call this dormancy. So spores will form, but cells become dormant. And with persisters, for example, if you catch them about 5 days into the stationary phase, you can revive them by adding a nutrient. We use butyl pyruvate. Other people use pyruvate, fructose, things like that. But then you start going beyond this. And the reality is that you come to a point where it is non-viable. But again, how do you define what non-viable? Because people are suggesting that there's always something that can activate them. And what this activation does, who knows? That's the point. But in reality, with certain pathogens, for example, persisters, they've demonstrated that you can't revive them. When you put them into a host, they revive. So maybe that's the definition. Can they cause illness? So it's one of those areas that people say, look, we've got enough to worry about. We don't need to worry about persister cells. But it just shows, what definition of death and things like that. So the ultimate dormancy is bacterial spores. And bacterial spores are resistant to heat, UV, chemicals. But can anyone remember what's in the core to make them resistant to UV, heat, and chemicals? Pretty silent. In the core, there's a glassy state. The glass is dipiclonic acid and calcium complex. So it's like a glass, DPA and calcium. It's got small acid-soluble proteins coating the actual DNA, locking it in times. The cortex maintains the dehydration. Then you've got spore coats on the outside, which is an impermeable layer from chemicals. So spores can obviously exist dormant for years and years, millions of years, but then germinate when they're exposed to germinating agents, L-alanine, for example. And it's quite interesting if you follow the thing. D-alanine inhibits the germination, whereas L-alanine stimulates it. Again, that's a clue on where life came from. I don't know, it doesn't not. But yeah, because the endospores are obviously significant, as we talked about, because some are deadly. They're very resistant to heat. So when we're sterilizing things like canning, we're inactivating the spores. And some are thermophilic, like Geobacillus, Stereothermophilus. That's used as a surrogate to verify the performance of canning. Our Cyclobacillus is another thermophile, which we encounter in heat pasteurized juices, because heating activates the spore. It says time to germinate, things like that. And then you've got Bacillus, which is facultative, aerobic, anaerobic, and obviously Clostridia, which are anaerobes. All right, so yeah, I'm dawdling a bit. So obviously, pathogens, when you think about food micro, is the most significant. And we know that, you know, in the 80s, well, you don't know because you weren't born, in the 80s, having all these outbreaks of listeria, having outbreaks of salmonella and things like that. And during the 80s, it's quite interesting that E. coli wasn't known about. Listeria just was discovered, say, a foodborne pathogen. So you could imagine how many we missed. But in 95, we had HACCP and a surveillance system. And basically, they were starting to look at, say, well, let's have a look at the prevalence of pathogens. And like I say, there's so many sort of assumptions made because there's only a fraction that's actually recorded. But the main one that's increasing, and it's not by total number, it's just proportion, is the Vibrio. So every year since 2000, early 2000s, every year we have this increase. So like you look at the graphs going into 2023, and you'll probably see Vibrio still going up. But why do you think Vibrio, of all the pathogens, is really increasing? The others are kind of flatlining. You haven't really gone high, but what is it about Vibrio? Oh, bless you. What's significant? Why would it increase?
Been going on since 2000s.
All right, fair enough. Now with Vibrio, there's a different, as always, different theories. One, obviously, people say it's climate change, you know, because it's getting warmer, and it's true to a degree. You know, we used to get Vibrio, obviously, in the Mexican Sea, where you go swimming and you get a cut, and before you know it, you've got your arm chopped off. But you're getting Vibrio now off Nova Scotia, off BC. The other part about it, obviously, we've got knowledge about the pathogen, and the third part is the diagnostics. Molecular techniques, because Vibrio is common to get VBNC state, a bit like Campylobacter in that respect. So it could be the case it's increasing. It's just a case of increasing detection. You know, as I was saying to Giselle, yesterday was on the radio, not radio, TV, talking about the salmon outbreak with oats. And essentially what's basically happening is that we're getting much better at detecting salmonella than controlling it, especially in things like oats and things like that. But anyway, to the point. So what we have is that certainly globally, it's still significant. We have this plateauing effect. So HACCP, you know, which I'll talk about on Thursday, was meant to be the safest food system where nothing can get past it, but obviously things do. But why do you think it is plateauing? Like, for all the efforts we do, you know, regulation, interventions, we can't seem to make a difference. And this goes for Salmonella, Campylobacter. You know, we bash you, we introduce stringent standards, but still people, why is that, do you feel? Not sure enough. So there's different theories. You know, one theory is that we're increasing a subpopulation. You know, like old people like me are more susceptible, and a high proportion of them. We're getting better reporting, you know, obviously. And yeah, people and the consumer is a weak link. The consumer is the one, or the food business, that do cross-contamination, that do temperature abuse and things like this. Anyway, to the point. So when we look at source attribution, what we find, obviously, most of the illnesses, cases, are caused by meat, poultry especially. Things like beef and obviously raw milk, if you go into that. Well, the most outbreaks are associated with fresh produce, because with meat and that, it's really down in a way to the consumer, the people actually preparing the food, isn't it? Because do they use a thermometer? Do they actually prevent cross-contamination? Whereas fresh produce, you know, as illustrated by the cantaloupe outbreak, we're really dependent on the farmer and the processor, who obviously did very wrong. It'd be interesting. I know what probably happened in that cantaloupe case, but I won't linger on it too much. But we'll describe the sort of pathogens. So the two main ones, bacterial ones, are Campylobacter and Salmonella. The number one cause of foodborne illness is what, do you think? So Salmonella, Campylobacter, they're up there, but what's it combined? All these sort of pathogens combined, which is the #1 cause of foodborne illness?
Is it salmonella?
Oh, no, someone else said, but what's the, you should know this, Jasmine, because you actually did give a lecture on it, I think.
I think I stopped before this.
Can't remember that. Yes, norovirus. So norovirus, all the bacteria, parasites combined don't outnumber. But as I say, with norovirus, you've got to say, well, is it only foodborne, which isn't the case, you know. But anyway, to the point, Campylobacter and Salmonella share the highest portion or highest incidence in terms of bacteria. And they're very contrasting. Campylobacter is this sort of weak bacteria, pathogen. You know, it gets inhibited and killed off by oxygen because it inhibits the enzyme pyruvate formate lyase, which is in its pathway. And it needs nutrients are provided to it. Very narrow temperature range, 42 degrees C being the body temperature of birds. And it's sort of spiral form, people suggest, so it can embed itself into the mucosa. Whether that's true or not, who knows? But it's an interesting fact. So as I say, it's very fragile. It doesn't really grow in foods. You're reading papers saying, oh yes, we've got compilers to grow, but they have to incubate at 40 degrees C into microaerobic conditions. So it doesn't grow very well. It's very sensitive to oxidative stress. It goes into this VBNC state very readily, and it's always going to be there at low numbers. But the secret with its success, though, is it's very virulent. So you need 500 cells to cause illness, and that illness is this explosive diarrhea you're going to get for a week, which is something to look forward to, isn't it? It doesn't really colonize per se. It kind of attaches and inflames the cells. And once you get, and it's, some people suggest it produces this toxin. And what this toxin does is open the fluid channels of the epithelial cells to get accumulation of fluid, and when you get accumulation of fluid, you're going to get diarrhea, and it kind of it's very explosive during that. So it's not very nice at all. And the symptoms, like I say, last for a year. You'll notice abdominal pains, and you obviously your immune system's kicked in, so you're getting fevers, aches, pains, chills, and all that. And it can last for days, sometimes for weeks, depending on how unlucky you are. So for a fragile organism, it's quite interesting that it doesn't grow very well, but still causes so many outbreaks. But the problem with it is these secondary effects. So the primary infection is explosive diarrhea, but the secondary infection kind of thing is the Guillain-Barré syndrome. So this is where, for example, the immune system responds to Campylobacter by producing antibodies. The antibodies bind, tag it for destruction by the macrophage. And so it's taken, but what happens is the antibodies also have complement to the gangliosides of our nervous system. The gangliosides, these junctions on the nerve cell. And so once the antibody tags them, it's basically tagging them for destruction. So you basically become paralyzed. It's not the cause, only cause, it's one of the causes. But as we'll talk about Salmonella, you know, that secondary condition is arthritis, septicemia, neurological conditions. Yeah, so basically with Guillain-Barré syndrome, the antibodies that are raised to attack the Campylobacter attack your nervous system as well. So it's a good idea not to get Campylobacter, I guess. So very few foods in actual fact, you know, poultry, obvious one, it likes to live in poultry, it's adapted, it's evolved to be in that niche, and it's adapted to cattle as well. So we'll find it in raw milk, only just a very small number of cells, 500 cells, which is very little. And in the Walkerton outbreak, for example, they had E. coli and also Campylobacter there. So the secret of its success is virulence. It's not its growth or anything like that. Then we come to the #1, which is, well, Campylobacter, Salmonella. You always interchange them for some odd reason. But Salmonella is very different, built to live in the environment. So it's like a tank. You know, it's adapted to a lot of different environments. DNA-wise or genetically wise, they're very homogeneous, Salmonella. You know, basically they share a lot of complements. So there's only one species, Enterica. Some people say, oh no, there's 2 species and things like that. And there's subspecies. Bengoria is the other species. But we can't just use that. What we have to do is define serotypes. So there's over 2,500 different serotypes, and these have different phenotypes. So they might have very similar genetics, but they've got very different phenotypes. And that's why you get some which are virulent, some which are associated with cantaloupe, some which are associated with enteritis, associated with poultry, and some with cattle like Dublin, for example. Now, typically then, there's, like I say, there's different virulence factors, and that really dictates what sort of infection it's going to be. So it's a bit complicated, this one. I'm trying to find the marker, but I can't. Oh, maybe not. Oh no, I've got it. That's good. So a traditional one is that you get your salmonella, you ingest it. Obviously, it's acid resistant, so it survives the acid of the stomach, and it gets to your GI tract. And then what it does, inserts a type 3 secretion system. And type 3 secretion systems, these tubes, as you know, which basically plug into the cell, a bit like our old modems, way before you were born, and they introduce a protein. This protein, in the case of salmonella, is called what? I'm surprised. All right, fair enough. Invasion protein A, isn't it? And what this causes, the rearrangements of the actin, which is our scaffolding of the cell, to ruffle. You know, a bit like when you go to the shop with a fresh produce, you try to ruffle the bags open in a bid for them to open. But it gives an opening, and with that opening, you go to this vacuole. This vacuole gives them protection. This is where the salmonella is going to start growing. So it can grow, but then go to, what it really wants to do is get into the macrophages. That's the key point. So it invades and it gets into the bloodstream. You get these dendritic cells, which basically capture foreign proteins and bring them to into the lymphatic system for destruction. So salmonella doesn't try to evade them. It says, yeah, let's go for it. Let's get into them. And their passage through there. You also get some salmonella to go directly to the lymphatic system. You know, basic lymphatic systems, this war ground. You've got your B cells, your T cells, your macrophages. It's there to clean the blood up. But and you would have thought the last place you want to be if you're a salmonella is in the lymphatic system, but it doesn't. It goes for it, a bit like Yersinia and Vibrio that way. So typically it goes into it, and there's some cells that kind of diffuse through the cell, endocytosis. So within the host cell, you have these vacuoles for transporting proteins and things. So it takes proteins from one side of the cell to the other side of the cell, you know, to do building things. And what the salmonella does in this case is hitch a ride. So it goes into this vacuole and just like diffuses through, very stealthy. But then you get some that are overexcited, and these are the hypervirulent strains. So you might hear about Salmonella Heidelberg, for example, hypervirulent strain. And what these do is that they get excited, so start expressing virulence factors within the host cell. And once you start expressing virulence factors, your immune system detects it, and therefore you start getting a natural killer cell. So the thing about salmonella, what it wants to do is lay low. It wants to get into the macrophage, neutralize that macrophage, you get taken to the spleen and things like that. But if you, a bit like the Squid Games, the stop go, if you activate it, you know, all hell breaks loose. So with the hypervirulent strains, you tend to get those that stimulate the immune system, causes dehydration, things like that. But ordinarily, the secret of salmonella is it once it gets into these macrophages, and these macrophages are designed to dissolve the salmonella, it neutralizes the macrophage and uses it as a vehicle to go transporting around the body in. And it just wants to become a host. It doesn't want to kill the host. It's got no intention of doing that. But sometimes, you know, things get out of hand, and it does. So the lymphatic system, like I say, Vibrio, Yersinia, a few other pathogens aim for the lymphatic system because it's like a highway to all where it wants to be in the liver, be in the spleen. So the thing about salmonella, as you'd probably guess, we've got the cantaloupe outbreak and now oats testing positive for salmonella. It gets around. And the secret why it gets around so many food types is that once, obviously, in the host, it's replicated, goes into manure. The manure is put onto land. The manure, and you get run off into water, and it can survive a long time, you know, years, well, to the point. So the traditional ones like poultry and eggs, you can understand, say, well, yeah, you know, it's associated with birds. The birds don't get ill with it. very hard to detect. So it gets onto the egg, it gets into the meat, and it gets into ground beef because cattle, even though you might think, well, no, cattle, salmonella, it's not quite the connection it does. Salmonella shed a small amount, so it gets into ground meat. But then it gets into things like cantaloupes, as you've probably seen, sprout seeds, and we sprout those seeds and things grow. Tomatoes is a common one. If you go down to Florida, for example, the tomato region, and sample the soil, you'll find salmonellas there, Montevideo, things like that. And obviously dry, low moisture foods. Because the thing about salmonella, once it's in the dry state, it survives, but it becomes very resistant to heat, becomes very hypervirulent as well. So all those things. So if you get peanut butter and things like that, you know what you're doing. So one that's not really high number, thank goodness, is Listeria. So the only reason to say about this series is responsible for most of the bacterial they call it undeclared milk protein or something like that. But then you get to the obviously the recalls down to contamination, and if you look at the stats, it's usually Listeria. So the last one was last month with spinach, because Listeria is everywhere. It's in the environment, it's psychotrophic, it can actually grow at cold temperatures like Vibrio, and it's different virulence. So you get something very virulent with go to France and if you're unpasteurized, the UK with listeriosis at the moment, well, apart from E. coli and things like that, due to the fact, and it's quite interesting that the incidence of listeria in France is much higher, apart from Scandinavia where they eat raw fish, but you can see the association, can't you? So there are 13 different serotypes of listeria, but the three main ones are things like 4B, 12A, 12B. It's not half a B, half an A. These are kind of linked to most foodborne illness outbreaks. And, there's this call from industry saying, well, look, we've got 13 serotypes of Listeria mono there. Let's just focus on these three and be fine. But, regulators don't like to take those sort of chances. Even when they get Listeria inocua, they start getting excited, things like that. So the thing about Listeria is that if you're fit and healthy like yourselves, you'll be fine, probably nothing, or you get mild flu. There's those high-risk groups of pregnant, immunocompromised that can get listeriosis, and that's where you get 30% fatality rates and things like that. So again, so these pathogens that you'll encounter, it's lots of regulations surrounding it. For that reason, it can be lethal. And the way it causes illness, like I say, is by invasion. So what it does is express lipases or phospholipases. These kind of erode away at the membrane. It goes into this vacuole and it grows in the vacuole, and it's got a protein called Listeria lysis, which breaks out the vacuolation system. So it zooms around the cell. If you go on YouTube, you can see these. So it goes from one cell to the next cell, which is the reason why it's got a long incubation period and things like that. This is Listeria. That's the Listeria there. This is active propulsion, zipping around the cells and things like that. But as I mentioned to you, if you're very unlucky, you get listeriosis, which is basically you can get stillbirths, you can get meningitis, septicemia, all these kind of conditions which can be lethal. And the foods reflect its environmental persistence. Basically in soft cheese, Listeria can get in from the dairy or the milk. With soft cheese, you're putting a certain amount of salt in it, which inhibits some of the bacteria, but Listeria likes it. Smoked salmon, if you go to any sort of smoking house, you get Listeria. Fresh produce, definitely. Who can forget the Jensen brothers of 2011? Who can forget even the Bluebell ice cream listeria outbreak? And deli meats. And interestingly, and some people say ridiculous, they actually found listeria on tea, dry tea, and they had a recall for that. Even though you say, well, who would drink cold tea? But anyway, this is the US for you, zero tolerance. So I'll go quick, like I said, I'm running out of time. So norovirus is number one, you know, hand to hand. The reason is that it's a handler's disease. There are outbreaks where we've had incidences where, for example, a dessert factory, they caused quite a few norovirus outbreaks because they had a sick worker coming into the plant and doing things. Clostridium perfringens, you know, it's a spore former associated with temperature abuse. So we just had Christmas. I bet a few people forgot to cool the turkey down, and they got Clostridium perfringens there. Staph aureus, again, this is as you know, a pathogen that thrives on temperature abuse, low acid products like cream and things like that. And it produces a toxin which is very stable to heat. So you can't destroy it. But it's this super antigen which stimulates your immune system. And you can get toxic shock syndrome where your immune system basically self-destructs. But the interesting one is toxoplasma, which is actually rated as, well, depending which stats you look at. Norovirus number one, number two is toxoplasma. And you might be thinking, well, I've never heard of any outbreaks of toxoplasma, but it's quite an interesting parasite because this is what parasites do. So toxoplasma, as you know, is an enteric protozoan. You know, it's basically lost its ability to survive in the environment, needs the host. And its natural host is cats. So the cats have this toxoplasma, they don't get affected by it, they're very happy, but cats do what cats do, and obviously they produce fecal matter. And what the toxoplasma does is have two kind of forms, the ois. The ois are kind of like spore-like form, no dormant, as in, but this is what the infective form is. Now, they also have these brandocytes. So these are kind of not dormant cells, but they have low metabolism. They're not vegetative, they're kind of in between, very slow metabolism. And they can be infectious as well. So the point is that for somehow, don't know how, obviously rodents kind of get a taste for fecal matter. They consume it, animals can. And this isn't a natural host for them, but what happens is that they get this sort of cyst, this brandocyte form, and this cyst embeds itself into the muscle, eventually into the nervous system. So the thing is that obviously we handle cats, we handle sort of meat and things like that. And when we ingest it, what the toxoplasma does is go into its vegetative state. It's starting to grow. But then the immune system detects it, and it goes into this what we call a cyst state, this brandocyte state, which basically is embeds itself into our nervous system, into our muscle. And what people are suggesting is that a lot of people are actually infected by it, but it doesn't do any harm because it's just laying dormant. But it affects our behavior, it takes us risk makers. So you might have seen the videos of mice which are infected with toxoplasma going up to a cat and saying, eat me, and things like that. And people suggesting the most successful people out there have these sort of toxoplasma infections. I don't suggest you do it as a career move because in reality, once you do get toxoplasma, you get this stillbirth, you can get organ failure and things like that. And there's a few others like Cyclospora, Cryptosporidium, and Giardia, also significant. But we'll see that another time. And to finish off on pathogens with Clostridia, yeah, this is the number one pathogen of concern. And anytime we have an outbreak, you'll hear it on the news. So these poor 3 Canadians who did the sardines, you know, they 3 of them died. And it's a very long road to recovery. And there's things like the carrot juice, where they pasteurize the carrot juice, obviously killing the vegetative cells, but waking up the Clostridium botulinum, which is resident in the soil. And there was a case where they temperature abused it, i.e. they left the cart at room temperature, and it was the highest level of natural, well, if you would believe it, naturally containing botulism toxin. And it caused 3 deaths. And I think the person spent 2 years in hospital and they died subsequently. It's paralysis, but people like Botox and that will inject it to get rid of wrinkles and whatnot. The nacho cheese one is where there was a gas station that was obviously gas station. You go to get your nacho cheese, maybe, and they didn't clean this sort of unit. You know, basically dust got in there. It became anaerobic, and I think it was 200 cases in that. Obviously, tin cans that lost the seals are an example. Vacuum packed fish, another example. Honey, you know, because obviously bees go around plant to plant. They can pick botulism up from the soil and things like that, put it into honey, and you get infant botulism. Yeah, because you pasteurize the honey to get rid of the spoilers, but you activate the Clostridium. And things like pulled pork, I won't go into that case, but it was about 4 years ago in Ontario. But we'll finish off with food spoilage. So obviously with food spoilage, the big thing about it, people think about food waste. And, you know, for a few years now, people have been saying, oh, I've got to get rid of food waste and all that. It's a bit of a fad, but, you know, we'll play along with it. Food waste and food loss are two different things. That it's food loss is loss up to the processor. After the processor, it's called food waste, and they're about equal, you know, even Stevens. And you might hear about videos where dairy farmers throwing away 30,000 litres of milk. You know, to say, oh well, you know, but we like food waste, but you know, they'll get rid of it for marketing. Anyway, to the point. So a high proportion of food waste is down to best before date. So people look at the best before date and say, oh, you know, it's day after, let's throw it out. And it's quite interesting. Every year, I get contacted by the press, not to say, oh, what fancy research are you doing? They're always saying, what about the best before dates, which is demoralising, but anyway, it's what it is. So people go by best before dates, but as you probably guess, it really depends on how it's handled. Now, what is the history of that product and how do consumers handle it? Because things spoil at different rates. So over in Europe, what they're doing is getting rid of best before dates. And you might think, well, that's good, isn't it? Well, not really, because it's putting all the risk on the consumers. And, you know, we talked about Clostridium botulinum. So what they're saying is vacuum packed meat, for example, saying, look, we tried it. We looked at the Clostridium botulinum growth. You're safe for another 10 days. Doesn't give you full of confidence, does it? But vacuum packed meat, as I say, is very high risk of Clostridium botulinum. But what they're saying is that, well, we know it's safe, but could you imagine eating a 10-day-old piece of meat? If you want to imagine it, just go and buy some today, leave it in for 10 days and look at it and say, it doesn't smell too good or look too good, does it? But, you know, this is what they're getting to the point at, saying, and all in the aid of food waste, which isn't good. You know, basically you are trading safety for quality. So what we really need to do is try to predict food waste best before date. So not just an arbitrary one, because it depends on how it's stored, it depends on the product itself. And so what can we do? So first of all, we have to define what spoilage is. And Maria, for example, this is one of her research projects. She's looking at ways to predict food spoilage. So when we think about spoilage, we're thinking, well, it could be off flavor, which could be true, production of metabolites. It could be the production of hydrogen sulfide or ammonia. It could be oxidation, polyphenol accumulation. It could be pectinases, degrading the fresh produce or visible growth. So the thing is, spoilage is a lot of different things, and it's very hard to put one sort of metric on it because, again, it's all caused by different modes. You can get chemical changes, you know, polyphenol oxidase or polyphenols accumulating. You get enzymes within the product, pectinases, proteases, lipases, auto-destructing, and you get obviously on top of that microbial growth. The ones that grow express enzymes and maybe make discoloration, you know, things like cyanophores in eggs. So the thing is, though, as you probably guessed, the mode of spoilage really depends on the intrinsic, extrinsic factors, especially when it comes to microbes. So under a certain condition, you'll get, well, the microbe has to be there, but the nature of the spoilage will depend on the microbe. And you'll just have to look at vacuum packed meat versus aerobic packed meat. Very different spoilage mechanisms. So, as I said, one of the big challenges then is we have to have accurate best before dates, but we have to try to say, well, what is the best before date? Like, and you can do it by century, you know, just temperature of use of product and say what it tastes like. You could use a chemical metabolite. This is where people are going. Is there a biomarker we can detect in real time almost to say this product's good or bad? And because microbes take long to culture. But what with the microbe we do at the moment is have a microbial criteria. And I get calls saying, I've got these counts of a product, does that mean it's good or bad? And you say, well, just look at it and see what it actually is. So we like microbial criteria because it's a number. It's easy tests to do, we know about it. But the trouble is, it depends on the food itself, depends on the microbe. With milk, for example, you can get very low numbers, but have big changes in the century, as you probably guessed. Whereas other things like spinach can have 10 to the 8 and still be good. So what we really need to do is that, microbial counts mean nothing. I've tried to tell this to people, but they never listen. What you have to do is find a specific spoilage organism. So this is obviously a microbe that's in very low numbers at the start, but then becomes dominant later on. It might not be the most dominant population, but it has the biggest impact in terms of product. So what we do to look at the specific spoilage organism is that we get a spoiled product, we isolate it, and then we do Koch's postulates. So who knows about Koch's postulates? Oh, you do, Jessica. I should have guessed.
Well, basically there's a set of rules, I guess, on how you can determine whether something is the cause of disease. So do you want me to list them?
Getting there. Well, I'll say I couldn't go over. So basically what you're doing is it's absent in a good product, present in a spoiled product. You can take the isolate, put it into a good product, and if it spoils with the same symptoms, you've got your spoilage organism. And that gives you, and the way you would do it is you'll have this combination of counts, sequencing data, also this sort of metabolomics now, or metabolomics being that trying to associate byproducts with microbes. You'll see a few papers coming out now that are promoting this, saying, oh, look, we found this Pseudomonas and we kind of correlate it to a certain metabolite, if you believe it or not. But anyway, you use a combination of methods and you get a specific spoilage organism. And then what you want to do is look at the actual byproduct of it. Is there a product there? Basically with Brofafrix, for example, I think it's 4-methylphenol. That's its main byproduct, causes spoilage. So you can measure it chemically because chemical analysis is much quicker than microbial and say, yes, this is spoiled or not. And with a specific spoilage organism, you can look at, say, well, how can we control it? Intrinsic, extrinsic factors, how can we predict its growth? Let's get a diagnostic test so we can go looking for where it's come from. There's a lot of benefits of going for specific spoilage organisms. And like I say, it helps you predict shelf life because you can say, well, this is going to be the main spoiler and we can get better shelf predictability on the actual. And if you get a kind of sensor in there, if you can imagine a sensor for 4-methylphenol within a packaging, then it can turn a different color when it's spoiled. It'll be probably I don't think anybody would want it in retail, but predictions. But there is this sort of go for shelf life sensors. This is what I say what Maria works on. And these really have to be inexpensive because you can't add much, but it's easier to look at a product to say if it's good or bad, rather than just have an arbitrary date. And if you ever go to embark on these, the best one to do is always fish. And the reason why fish is good, because it's got 3 modes of spoilage: autolytic, chemical, microbial, but the products are all the same. So typically, like when somebody comes to me and says, I've got this spoilage sensor, and you try to impart on them saying, well, you can't do one for all products. The best one to work on is fish. And that's why most of the sort of spoilage sensors focus on fish, TMAO and things like that. Do you want me to go to the end, or do you want me just to circumvent? Because I know I'm over time now as it is, isn't it?
Let's take a vote. Are you okay for another 7 minutes? To finish off until slide 89.
I would vote for saying, let's get out of here, isn't it? All right.
See to the end.
I'll keep going then. Yeah, don't worry if you want to leave though, it's recorded, so you can get the last bit. Anyway, to the point then. So spoilage of fish is if you're going to develop a sensor, this is what you want to develop, because regardless of the mode of spoilage, the end result's the same. So we have TVB, total volatile nitrogen. We have TMA, total trimethyl acetate. Amine, trimethylamine, I'm getting more. Free fatty acids, thiobarbituric acid. The K value is the one that people are putting their sort of hat on. So the K value is really dependent on the degradation of ATP. So all cells of ATP, regardless. And it goes through degradation products. So we'll talk about on the next slide, but it's called the K value. And you can use the K value to correlate. You get biogenic amines, decarboxylated amino acids, and these have health impacts. These cause the kind of allergic reactions and things like that. And obviously the count as well, which doesn't mean that much. But the main one people are looking at is the K value. So ATP is found in all cells, and obviously when we die, these start getting degraded, and it goes to inosine monophosphate, inosine and hypoxyxanthine. So it goes without reason, doesn't it? If you've got a very high concentration of IMP, inosine monophosphate, you're at the early stages of spoilage. If you've got hypoxyxanthine, you're pretty well at the end of spoilage, isn't it? So people can use the K value because, like I keep saying, chemical tests are much quicker, easier to adapt than microbial sensors. And these are examples of tolerance limits, because obviously some are more obvious than others. So total volatile basic nitrogen, TMAO, or TMA, which is the one that's used to be used. But you'll see, I say, the K value in a few sensor devices. And TMA was the first one because people noticed they could correlate the shelf life of fish to this TMO, which is a breakdown product, as we talked about. So it breaks down naturally or microbial action, and it's accumulating. I'll tell you, saying, is this fish fresh or not? So if we did TMA in our fish in theirs or a supermarket here, obviously it's going to be very high because we're so far from the sea, and that fish is probably a bit aged. But you can use a simple test, instant results saying, I'm going to accept this fish or reject it. Put it on to 30% reduced thing as it is now. And visibly though, you can see, if you're an expert, you can see fish degrade. The mucus goes all sticky and starts getting an odor. The eyes get sunken. The proteolytic breakdown of them. But as I mentioned to you, we're not all fish experts. You want a technician that says yes, good or bad, based on a number. So as I mentioned, the spoilage process, a bit like other spoilage processes, is all down to the chemical reaction, the enzymatic action, the microbial. That they tend to be overambitious and try to do everything. Well, it doesn't quite work like that. But people are starting to develop sensors now. Artificial noses have been around since the 1980s, where people try to recreate the nose by having these arrays of polymers. I worked on this in Manchester all those years ago, and there used to be one called the Bloodhound, original name. But the thing was, it's so insensitive that you had to, like, if you were detect diacetyl, it was overpowering you, and you get a small signal on your sensor. But I think they've come some way since then. But there's nothing as sensitive as our nose and things like that. But there's other things like detecting ammonia, detecting sort of pigments. And I think what Marie does is she looks at spectral analysis. So she's got AI technology, well, she'll call it AI now, to say, yes, this product is this time. And it won't be a spectral analysis in the package. You'll have these sort of radio receivers and transmitters, which you put in the packaging, and then it will send a signal back to a supercomputer to tell you if it's good or bad. Again, retailers don't want this. They might say, oh, yes, it's the best idea. But when it comes push comes to shove, they tend not to. For many years, there's been this temperature signal, for example, temperature sensor, which is just a sticking label. You put it onto the pack and it turns red when it's being temperature abused. Retailers didn't want anything to do with it. And the reason, as you probably guessed, they don't want a whole row of their product looking red, have red dots on, no one's going to buy it. They always say, isn't it, it's a bit like when we go to get our 50% off, you know, what can we push? What can we give the consumer when they'll buy it? Because once they've bought it, we don't really care if it spoils the next day or anything. Yeah, Costco are good for that. Anyway, the final force, you know, hopefully we've come through this pathway of evolution and we've decided that we do live in a microbial world. Microbes have adapted, they're adaptable, but understanding how they evolved and what their evolved, what niches they inhabit, gives us an idea of pathogen food associations, gives an idea how we can control them, and even gives an idea how we can cultivate them. You know, do they need high salt? Certainly foodborne illness is one of those things that are ongoing. We don't seem to be making any inroads, and likelihood is we'll get more outbreaks like the cantaloupe one, over 400 cases, 10 deaths and things like that. But where the money is shelf life. Like when I go to industry and say, I've got this method for decontaminating eggs, for example, you get blank look. You say shelf life extra 2 days, suddenly you're the best friend. So when you're doing research, even though there's benefits of the shelf life, then, but yeah, the real benefits of shelf life, that's what you sell. So if you want to read, do some background reading, these are free downloads. The Bad Bug book is the pathogen one. You can get a book on spoilage and things like that. And just to whet your appetite and get you excited, on Thursday, we're going to talk about food safety.
Wednesday.
Oh, I'll make a note of that. Wednesday. Yeah, just come and knock on the door if I'm not. So on Wednesday, we're going to talk about food safety regulations and modernisation and things like that. Oh, that's good. So if you've got any questions or anything, or? All right, fair enough.